Mørkvedbukta in Bodø, most of the year it's ravaged by strong winds and rain, and even though it doesn't seem like it, the area is filled with life, both above and underwater. Where one group of animals stands out, in the sense that it hasn't been studied as much as others, which leads to unanswered questions, which we will try to answer on this journey to unfold the mysteries of the genus Littorina. The initial question that needed to be answered was, what do we want to find out about these snails, and what can we expect to find? So firstly, we had a talk with Dr. Anja Westrom, evolutionary biologist who has studied these snails. After a digital meeting with both her and Jus Reimarka, our teacher, we came to the conclusion that we have 4 species of snails that we can expect to find in Mørkvedbukta, which can be Littorina saxatilis, arcana, Littorea, and obusata, or flat periwinkle. The flat periwinkle is still under discussion whether it is Littorina obusata or another species. So for the sake of this project, we will refer to it as flat periwinkle. The only problem we faced was that Littorina saxatilis and Littorina arcana has very similar outside morphology. This meant that we had to bring samples back to the lab and dissect them in order to differentiate the 2 species, compared to the species Littorea and the flat periwinkle, which had clear morphological differences. The Littorina Littorea is easy to identify because of the angle between the aperture and the main shell, which is steep, compared to the Littorina arcana or saxatilis, where the angle is flat. The flat periwinkle species is easy to identify by its flat shell, which doesn't come to a point as the other 3 species do. The first location, Zone A, is a south-facing area which is sheltered with a lot of different brown algae with a narrow aperture to the sea, creating a tide pool which serves as a shelter for the different snail species among other animals. Zone B is also south-facing with a constant slope. It's not particularly protected but not exposed either. The substrate is made up from large pebbles and rocks covered in different algae. The amount of seaweed decreased the further from the waterline we sampled. Zone C is very protected with a constant low slope with muddy substrate, very different from the other zones. The first sampling contained significantly more seaweed than the other 4. Zone D is a very good example of an exposed area, which we expected would be a defining factor for our sampling. The substrate is very rocky with little to no seaweed covering the area, providing no shelter for the snails. Zone E consists mainly of a narrow canal with a constant slope, with a substrate of large rocks and seaweed that decreased with every sample, further from the waterline, the same as Zone B.
Here we are at the first sampling day, in Zone B. At the beginning, recognizing the different species was difficult, but with time it became faster and easier. The different areas we chose show many differences, giving great variety to the quadrats. Here, for example, we have a completely rocky section, elevated from the tidal zone. Here you can see the process of moving to the next quadrant in a transect, measuring the distance from the new position from the previous one, to build an indicative distance from the waterline. We tried to get samples showing different slopes to get a more representative data. Well, it was not always easy to collect those snails.
Some of the quadrats were even partially or completely submerged, giving us more chances to observe different behaviors and other cool animals, such as small crabs or flatfishes. Our hypothesis is based on initial observation from the first sampling, which was at the end of September, where we discovered that the Saxatilis arcana snails were located furthest from the waterline. The snails we suspected to be Saxatilis or arcana we brought back to the lab. Based on the observations, we can assume that the distribution of the species will vary based on the difference in substrate and distance from the waterline. So from this, we can make a hypothesis which says that there is a correlation between the distribution of the species and the tidal zones or the distance from the waterline. So from this, we tested the hypothesis on the different low sampling locations.
This is the last day of sampling. So we had like last week and a week prior to that, we sampled from the areas A, B, and C, and we already processed those snails. And now we're sampling from location D and location E when we're finished here. When we go sampling, we make sure that when we pick the snails, that we don't throw them around outside their local ecosystem. Inside this square or this quadrat, we keep them afterwards, so we don't throw them into water or other places. So we keep like the distribution of the species and the location. We go through this chart over here, where we write down the coordinates of each quadrat, and then we take pictures of the whole quadrat as a whole, and then we take pictures of each smaller quadrat to get an overview of the sampling we're doing. Then we write down the time so we can check later on online how far up the water level is for each sample. Then we take an estimation of how big the slope is, so we can check later on in the maps how big the slope actually is. And then we take a quick assessment of the area, whether to I don't know, describe the substrate, or if it's a lot of seaweed, if it's very rocky, or if it's the large boulders we're collecting from. And as well, for this first sample, we we measure from the waterline up to the first sample, and then we measure 2 or 3 meters, give or take, between each sample. And we have 5 samples for each location, and we have 5 locations. The last thing we do is that we sample all the snails that we think are the Saxatilis or arcana species, and then those will collect and bring back to the lab to identify the different species. Another thing we do is to pick other snails, if there are other snails, from the species Littorina, or the flat periwinkle ones, which are in discussion with what kind of species it is.
To store the snails until the lab days, we just put them in the fridge in the lab in Bodø, where at around 4 degrees Celsius they can survive up to 2 weeks.
Here we are determining the size of the snails by measuring its width and length. The width is measured from the end of the opening in the shell to the broadest part on the opposite side, while the length is measured from the top of the shell to the bottom. Then we report the measurements to be written down.
Each snails deserve a mugshot. Before extracting them from their shell for identification, we took a picture of every individual with its ID code. This came handy in moments where we misplaced some data. We could also check the measurements from the pictures.
To initiate the dissection, we first generate a hole into the top of the shell with a pestle. Then we take the forceps and push the body out of the snail from the aperture or the natural opening through the hole we just made.
When we determine the gender and species of the samples, we use 3 categories, and those are Arcana, Saxatilis, and male. The female Arcana is identified by a white jelly gland and embryos without shell, while the female Saxatilis is identified by a broad pouch and embryos with shell. We do not have enough knowledge to determine the difference between male Arcana and Saxatilis, so the gender identification is purely based on genitalia.
After analyzing the species and gender of the snail, we will cutting off its head and put it into a tube with ethanol. The head must be not contain any eggs or embryo to avoid being contaminated with DNA from different individuals. And then we need to clean all of the equipment that we used before doing the next one.
So this is our R script. First you load the datasets and the data you need to analyze, and then you proofread it with showing a few lines in the console. For the first step, we want to have an overview of the distribution of the species based on the distance from the waterline, and we can already see the different preferences of the species. For more detail on the individual species, we follow the same parameters, but for each species adding regression lines for every location. Another interesting data to look at is the size distribution in the different locations to compare with morphology of the different areas. Immediately we see that location D has smaller snails than the others. This can be explained by the area being extremely exposed.
Each dot represents a square or sample area, while each line represents one of the 5 locations we sampled from. Black is location A, Red is location B, green is location C, blue is location D, and light blue is location E. For L. littorea, we can see that the number of individuals increase in every location except location C. We believe this is because this species does not like to live in the flat, muddy area, but instead prefers a rocky habitat. Arcana, similar to Littoria, had an increasing number of individuals in every transect except one, though the two species differ in which transect they decrease. In Arcana, transect E is decreasing. This may be because we sampled later in the day, when the tide was higher. Lastly, the distribution of Saxatilis has very varying results. Two transects, B and C, are increasing. One of the transects, A, is almost flat, while two, D and E, are decreasing. In the graph for obsata, or flat periwinkle, we can see that the transects decrease with the distance from the waterline. This made us believe that this species is more comfortable near water.
From the look of the results of the sampling, we can clearly see a link between the distribution of the different species and the distance from the waterline. This shows that the hypothesis we presented earlier can be proved by the estimated and educated guess that there is a link between them. The species Littorina, Saxatilis, and Arcana were found to prefer the shore or further from the waterline, in different cracks and big rocks, compared to the Littorina littorea, which we found close to the waterline, surrounded by a lot of seaweed and other abiotic factors, as well as the flat periwinkle one.